So after the last lecture, there were some questions regarding the ELISA technique. Remember, we talked about in our ELISA, we can have either what we call a direct assay, indirect assay, and sandwich ELISA, right? So for direct and indirect assay, we coat the ELISA plates. Okay, so this is a typical ELISA plate, like this. Okay, so again, I talked about how we can have 96 wells altogether, 8 times 12, right? So it is a system like this, and each one of these well actually is inactivated, meaning that you can cross-link to a protein. So if you put your antigen in these wells, then the antigen will be coated onto the well. And then the only difference between the direct assay and the indirect assay is that in the direct assay, you have the primary antibody already conjugated to an enzyme so that you can detect the presence of the primary antibody and hence the antigen by putting in substrate into these wells, okay? And for indirect assay, you don't have a primary antibody conjugate with an enzyme, but you detect the primary antibody with a secondary antibody. So most students ask me why you want to do that. Okay, so imagine that in my lab, I have to do 10 different ELISA every day, okay, and or EIA or any antibody techniques that require primary antibodies. And if I have to conjugate my primary antibodies in all the assays, my whole lab will spend all the energy doing that, because it is a difficult experiment. And after you conjugate the enzyme with the antibody, it takes time to test out whether your primary antibody working or not, right? So it takes a lot of time to do that. So instead, nowadays, from different companies, you can purchase secondary antibodies that can recognize the primary antibody. So in my lab, for example, I have secondary antibody that is raised in mouse, that is raised in goat, that is raised in horse, that is raised in rat. That is already conjugated to an enzyme, like horseradish peroxidase, which is the enzyme that you will use in your lab session, okay? So all I need to do is to find out where this primary antibody is being raised. Okay, so if it is raised in rabbit, then I will use an anti-rabbit secondary antibody conjugated with an enzyme. And then I can do this ELISA right away. You see what I'm trying to say here? So it is for convenience. And in your example, like the pregnancy test that I talked about in the last lecture, it is a commercial, it is business. So they can mass produce huge amount of the color primary antibody for detection. And in that case, it is much easier to have the primary antibody conjugate with a color or enzyme. So they can use it all the time. Okay, so in that case, they can mass produce a primary antibody already conjugated with an enzyme or a color for detection purpose. But in the laboratory setting, it is much easier for me to buy all the secondary antibody, conjugate with an enzyme, or conjugate with a 404 for fluorescence detection. Like again, detect green color, yellow color, red color in the immunohistochemical staining, immunohistofluorescence staining or immunocytofluorescence staining. So it's much easier for me to do that. Is that okay? Okay, thanks. And in the last lecture, we talked about how we will talk about SDS page. In the end, towards the end of the last lecture, we talked about today's lecture focusing on SDS page. And SDS page is to separate proteins based on a solid, based on a acrylamide gel, polyacrylamide gel. Okay, so it is very much like agarose gels you have learned from Professor Lui. Instead, a polyacrylamide gel is capable of separating smaller molecules. DNA molecules are much bigger in general. And protein, say, depending on the size, right? So some protein like can be as small as 100 kilodalton. And bigger protein may be 300, 400 kilodaltons. But still, we're talking about smaller size. Comparison to DNA, like 3 kb, 4 kb of DNA is much bigger. So we can use agarose to separate them. Polyacrylamide gel is therefore very important for separating smaller molecules, such as protein. And after polyacrylamide gel electrophoresis, we'll talk about how we can combine this technique with antibody detection, and that is called Western blot. Western blot is used routinely in all the biochemical laboratories. My lab perform Western blot on a daily basis. Every day we do Western blot. So it's a very important technique. And then we'll talk about isoelectric focusing. This technique is comparable to the isopycnic centrifugations. Very similar kind of thinking. Okay, so we'll go through that. And then we'll combine all the techniques together, meaning SDS page, isoelectric focusing, to do a two-dimensional gel. And then you can also do Western blot on a 2D gel. Okay, so this is a typical setup for the SDS page. So you have an upper reservoir and a lower reservoir here. And this is where you have the polyacrylamide gel set in between two plates, forming a gel slab. And then, as you can see here, you have the wells that you can use combs to form the wells when you set up the gels. And then you load your sample into these wells, and then you hook up this whole apparatus to a power pack. And the positive pole is on this side, the anode is here, and the cathode is here. So proteins that are labeled with a negative charge will then migrate down and being separated in the resolving gel. There's one more thing I'd like to mention here. So in this particular setup, you have two sections in the gel. One is called the stacking gel, and the other one is called the resolving gel or separating gel. And these two regions of the gel serve two different purposes. Okay, so stacking gel functions to pack all the protein into a tight band so that you can increase the resolution. When the protein in very tight band, then the resolution of the gel is increased, right? That makes a lot of sense, right? The second thing is when you have all the protein packed into a very tight band, then suddenly the sensitivity of the gel is also increased. You don't need so much protein. but you can still see the protein by staining the protein with Kumasi blue later on. So before we go about talk about the principle behind this separation, remember, first of all, remember that the stacking gel is to stack all the proteins together into a very tight bed. And this is achieved by a discontinuous buffer system, by changing the pH along this whole system, then you can achieve that. And we'll go through that later on in today's lecture. And after the protein has entered into the resolving gel, suddenly there's a change of the condition of the pH and things. There will be a constant voltage gradient along the resolving gel, just like the agarose gel, just like normal gel electrophoresis. The proteins will be resolved based on the properties of the proteins, meaning that the protein will be resolved based on the size of the protein, size and charge of the protein. Okay, so the velocity by which a protein migrates in the separating gel or resolving gel is dependent on Q over R and E over D. Okay, so Q is the charge of the protein. R is the radius of the part of the protein. And I'm sure you have learned that equation from Professor Lloyd already. So it's just like electrophoresis. So the protein migrates in the gel. The speed at which this protein migrates is related to the charge, right? So the more negative charge the protein is, then it will migrate in a faster rate. If you have the same protein, if you can attach more positive charge, then you can migrate at a faster rate. But it's inversely related to the size of the protein. You can imagine the protein gel. It's like this classroom here. You have all the seats here. If you have a protein of the size of my fist, then the proteins can squeeze through the gel very easily. But if you ask me, a bigger protein, to go through these chairs, to go to the back of the classroom, it will be very difficult. It takes much longer time for me to do that. I can still do it, but it takes much longer time. Okay, so this is the principle why it is inversely related to the size of the protein. And then it is also related to how much power you give the protein to go through this classroom, right? So this is the voltage, the electric field that you apply. So the power pack, you can run it 100 volt, 200 volt, or 300 volt, and then the protein migration rate will be dependent on that. And also the distance between pole. And we're talking about, therefore, E over D is the voltage gradient. Okay, so you have 100 volt per 10 cm, and 100 volt 10 cm, then you have 10 volts per cm. So that is the voltage gradient along the gel. And this is the determining factor for the protein migration velocity in the gel. So make a lot of sense. So it's like you're pressing the gas pedal in a car, right? The more gas you put in, the faster it will go. It's the same as that. Okay, so this is a very simple relationship that we're talking about. V related to Q over R and E over D. And then because of the fact that we can use the acrylamide gel to separate protein, then you can use it for protein size comparisons, right? So you can see that this is a standard marker lane with molecular marker. So you know the molecular weight of all these proteins. So after you run the protein in the gel, this is 18 kilodalton, 29, 43, 68, and 97. So you know the molecular weight, and then this is your protein of interest as an example. So you can actually plot a graph, distance versus molecular weight, using these molecular weight marker, and then measure the distance traveled by your unknown sample, and then you can guesstimate, you can estimate the molecular weight of your protein of interest like this. Right? So we are going to talk about protein size comparison, and we're going to talk about using reducing agents and using detergents, giving rise to reducing, non-reducing, denaturing, non-denaturing gels. And for denaturing gel, basically you use SDS. And for reducing SDS page, then you put in, I talked about in the last lecture, some reducing agents, such as beta-mercaptoethanol or DTT, dithiothiazole. We talked about that last lecture. And then the tight band protein band is produced by the discontinuous buffer. So we're going to go through all these different items in this list in the lecture. So I talk about why use ACO-amide gel to separate proteins, because it gives rise to a much tighter matrix for the protein to be separated. And this is acrylamide, and this is this is called BIS. Basically, it is a dimer of acrylamide, so it functions as a cross-linker. So if you have only acrylamide, and then if you polymerize that, it only form a chain, a string of acrylamide, polyacrylamide, and it will not give rise to a very rigid structure. So therefore, you have to put in a cross-linker, this allowing the formation of a bridge between strings of polyacrylamide. Okay, so you can imagine that this will form a small pore, as you can see here, for the protein to squeeze through, and another hole here, another hole here, and then it forms a very tight matrix for separating even very small protein. You can also adjust the percentage of acrylamide. So if you are separating bigger proteins, then you use a higher percentage gel, like a 15% acrylamide for smaller protein. And if you are separating large protein, you can use an 8% polyacrylamide gel that has a much bigger, these pores being generated, and then you can use it for separating large proteins. Okay, so this is just to show you that DNA on average is much bigger than protein. So nucleotide pair, 649 Dalton. So 1 kilo base pair is 649 kilodalton. And this encodes 333 amino acid, which is the size of 36 kilodalton. So DNA are much bigger. Okay, so this slide illustrates how we can perform reducing, non-reducing, denaturing, non-denaturing gels. Okay, so the first thing is SDS. It's called SDS page if you put in SDS in the sample buffer. So SDS, sodium dodecosulfate, has a structure like this. So you see the carbon aliphatic chain, and then there's a sulfate group, and then a sodium ion here. And so dodecosulfate is negatively charged. So if you put SDS into the sample buffer, and then the sample, you put your sample into the sample buffer, and then you boil the sample, the protein will open up when you boil. When you boil, the protein opens up, and then the protein containing a lot of hydrophobic amino acid residues, right? So it will interact with the carbon side, the carbon chain here, And then the protein will be labeled with negative charge. And then you can separate all these proteins in the SDS page because of that, right? So remember, the end node is on the lower solution buffer, right? So the protein now is labeled with a lot of negative charge, will travel down the gel. And in addition to that, you should notice that the charge density of the protein is the same. So the longer the protein, the more charge it will have. The shorter the protein, it will have less charge. So now the charge of the protein is taken away from the equation. Velocity is proportional to charge over size, right? And then the charge density of the protein is removed from the equation. And then therefore, the SDS page can be used for estimating the molecular weight of the protein, because only the size that determines also the charge and also determines the rate at which the protein travel in the gel, right? So only the size that determines the velocity now. So SDS page is therefore a good estimation of, it's used for the good estimation of the size of the protein. If you take away SDS, you can still do it. But neutral protein, positively charged protein will not run into the gel. Only negative charged protein. And also, the charge of the protein will be different among all the proteins. Therefore, if you run a natural gel, then non-denaturing gel, sorry, then as a result of that, it is both the size and the charge of the protein that determines the position of the protein after you run the gel. Okay, so remember that. So we also talk about in the sample buffer, we have, if you put in beta-mercaptoethanol or DTT, then you can run a reducing or non-reducing gel. Okay, so reducing gel is you put in this reducing agent. So this is a protein that has two subunits interlinked by a disulfide linkage. And these molecule, DTT or beta-mercaptoethanol, is able to reduce that, to break that disulfide linkage, to form the sulfhydryl group separately from each other. So when you run the gel, then this protein will form two different bands because you have already reduced them into two polypeptide chains. If you don't put in the DTT, then you will see one bigger protein running together in the gel with two subunits. Okay, so this is reducing and non-reducing. The sample buffer also contains glycerol because it will make the sample heavier and load into the well easier. It contains also bromophenol blue, which is a dye, a blue color, so you can actually monitor the progress of the electrophoresis process. And you will see that when you do your laboratory. So after electrophoresis, the proteins can be stained by many different techniques. The easiest one is by using a dye called Kumasi blue. Kumasi blue will stain all the proteins in the gel, and then you, after a repeated process of destaining, taking away Kumasi blue from the gel matrix, from the polyacrylamide gel, but without the protein, then you can see the blue bands on the gel, and each one of you is will run one protein gel based on that. Huh? Oh. Okay, so I mentioned how the SDS page can be used for size estimate, protein size estimation, right? So if you plot the mobility, that is the position of the protein band with respect to the beginning of the separating gel, and compare that to the molecular weight in log size, empirically, experimentally, you will see that you will get a straight line like this for some unknown reasons. And then you can use it, you can plot a curve like that, and then use it for the identification of your unknown protein in terms of molecular weight estimation. Okay, so this is a different types of SDS page or silver stained SDS page. We can see the molecular weight of some standards alongside, as you can see. Okay, so we talk about denaturing, non-denaturing gel. We talk about reducing, non-reducing gel, right? So now we talk about SDS page using a discontinuous buffer system. And this is why you can stack the protein together in the gel. Okay, so I used to use a overhead projector to illustrate that, but now because I have to record the lecture and I don't know how to do it. So I'll try to verbally describe that, but it's a difficult concept. So before you can understand SD discontinuous buffer, first of all, I describe the system to you. So discontinuous buffer, meaning that there's a change of pH along the gel, right? Buffer is the pH. So the upper reservoir contains glycine, pH 8.3, using tris as a buffer. So it maintains a relatively stable pH of 8.3. And also in the lower reservoir, same buffer. Remember that it contains only glycine. Okay, glycine is the running ion. So when you apply a voltage gradient across the gel, then the glycine at pH 8.3 is negatively charged. It will run into gel as the running ion. And then afterwards, in the stacking gel, when you mix the solution to produce the stacking gel and the separating gel, you don't put in glycine, but you use chloride as a running ion. Okay, chloride is the running ion. So when you apply a voltage across the gel, then the chloride ion will go down. And then the stacking gel, the purpose of that is not to separate the protein according to size. So we use low acrylamide percentage, only 3%. The purpose of the stacking gel is to stack all the proteins together into a very tight band. In the separating gel, you can have 8 to 25% or gradient acrylamide depending on the size of your protein. So you can run an 8% gel or a 15% gel or 25% gel, or you can run a 8 to 15% gradient, 15 to 25% gradient, depending on the molecular range you'd like to separate. Okay, so another thing is that the stacking gel has a pH of, the stacking gel has a pH of 6.8, which is the isoelectric point of the glycine, meaning that glycine, if entered into this stacking gel, suddenly the glycine will lose its negative charge. So in a population of glycine, the average charge of the glycine is 0. Some may be positive, some may be negative, some contain a positive and negative. Okay, but the average is only 0. So glycine, after enter into the stacking gel, suddenly there's a charge change, and it will lose its mobility. In the separating gel, however, the pH is 8.8 again. So once the glycine has entered into the separating gel, it becomes positive, negative charge again. And then it will migrate like normal electrophoresis. So what is happening is that in the stacking gel, you have therefore created a moving boundary above the moving, so this is the moving boundary here. Above the moving boundary, you have the glycine that has a slower mobility. Below the moving boundary, you have the fast leading anion, which is chloride. And protein has an intermediate mobility. As a result of that, protein will get stuck in the moving boundary. And the moving boundary will slowly move down the stacking gel, collecting all the proteins and pack them into a tight band. And once the protein enters in the separating gel, there's a change of pH again. And glycine now is free to move forward. And there's no more moving boundary. The proteins will experience a constant voltage gradient, and it will be separated according to size and charge, like normal electrophoresis. Is that clear? Okay, so you can imagine that in the next slide, this is what is happening. So if you can generate a moving boundary, above that is glycine, below that is chloride. Imagine this, that now this classroom is the stacking gel. And you, each one of you is one protein. Now you scatter along the stacking gel. And the moving boundary is me. Okay, so I apply a voltage across the gel, and glycine enter into the gel. Glycine loses mobility. Glycine is behind me. Chloride is in front of me, and I'm the moving boundary. And as electrophoresis proceeds, then this moving boundary will also go forward. And then I collect one protein here with me and pack that in my moving boundary and collect two. And as I go along the gel, I collect each one of you, originally scattered in the classroom. But now you are all packed inside this moving boundary. Is that clear? This is what is happening in the stacking gel. So this diagram, although look rather primitive, explains exactly what is happening. Okay, so in the stacking gel, you have this moving boundary going down and collecting all the protein into a very tight band. And you can ask, why is that the proteins are not behaving like how they behave in normal electrophoresis? And it is because in the stacking gel, you have generated a discontinuous voltage gradient in addition to a discontinuous buffer system. Because you can imagine that behind the boundary, you have glycine that has no charge, right? Very high resistance. And in front of that, chloride has charge, low resistance. And according to Ohm's law in physics, you know that the part that has a higher resistance will take away most of the voltage. Right? So imagine that just for illustration, it has no real meaning to this analogy. So imagine that you apply 100 volt here, and then this is 9 ohms, this is 1 ohm. So this region here is 90 volt, and this region here is only 10 volt. And this is why the protein behave differently compared to in the separating gel where there is a constant voltage gradient. So it's always 10 volts per cm, right? So the proteins will therefore behave like normal proteins in an electrophoresis scenario. But in this case, because of this continuous buffer system generating a moving boundary, and that result in a discontinuous voltage gradient as well. Therefore, the proteins are behaving differently. And you can only explain how the protein move along the gel according to what I've just told you. Is that clear? Okay, so you go home and think about that. It is a rather interesting concept. Okay, but what I'm saying is that we can play around with the pH in such a way that we can therefore control how the protein move along the gel. Okay, so I spent hours generating this diagram for you. So in the beginning, so this is a stacking gel. This is a separating gel. So you load your protein in the well, and then you apply the, you hook it up, you hook the system up to a power pack, and then you apply a voltage field across the gel, and then glycine start to move in, and then chloride in the front, and then all the protein will stack in the moving boundary going down until it has reached the separating gel, and then glycine will move into the separating gel. Suddenly, there is a change of buffer. Glycine now is negative charge, and there is a constant flow of ion across the gel, a constant voltage gradient across the gel, and the protein will be separated in this separating gel, 8%, 12%, 15%. And then it will be separated according to size and charge. But if you run SDS page, it will be separated according to size only. And then you stop the gel here, and then you can stain the protein with combustible. This is what is happening in the electrophoresis. Is that clear? Good. And if you have problem, you can, there will be a tutorial next week. Today, I try to finish all the lecture material, and then you go home, digest the material, and then next week, we can have a tutorial. You can ask me any questions. Okay. This is a difficult diagram for you to digest, but this is one of your lab. Okay, so in the lab, you'll be given a bacterial culture. And you spin down the bacterial culture. The culture, the bacteria, we use it as a small little factory for making recombinant protein for us. So in the bacteria are infected with a plasmid. Plasmid is a circular piece of DNA. In this case, it's double-stranded. Or you can use a bacterial phage. But in this case, plasmid is better because plasmid can actually replicate in the bacteria and go up to like thousands of copies of the same plasmid in one bacteria. So one bacteria, therefore, is a small little factory that produces a recombinant protein for you. And how does it produce the protein? It's by transcription and translation. You know this process, right? So this is a plasmid vector that you can buy from companies. Okay, this is routinely used in my lab. It's called PRZ8. And there are 3 different reading frames, right, in translation process. And for the ease of cloning, we have 3 different vectors because of that, each with 1 DNA base pair difference. After just before the multiple cloning site. So You have PR set A, PR set B, and PR set C, 3 different reading frames. But in your laboratory, you will use PR set A. This is the vector that you use. You can see that this double-stranded circular piece of DNA contains an ampicillin resistant gene. So all you need to do is put in ampicillin into the culture, and it will select for the plasmid containing bacteria. Other bacteria will not grow in that culture. Okay. It contains an original replication, meaning that the plasmid is capable of replicating in the bacteria. Then go up to several hundred to up to 1,000 copy number of the same plasmid. So this is now a clone. So all these plasmids contain the same DNA material. So it's a clone. It contains this stretch of DNA regions. Okay, so it contains a promoter. A promoter is called this promoter is called T7 promoter, and meaning that this promoter is driven by a T7 RNA polymerase. T7 RNA polymerase. Okay. And then after the promoter, it has a RBS, a ribosome binding sequence. So it ensures that efficient binding of the ribosome in the bacterial setting to the transcript once it's being produced. It contains an ATG initiation codon. So this is where the translation will start. ATG is this translation start site, right? So ATG, after that is 6 histidine tag. 6 histidine amino acid will be translated at this region. And this is important because now you can use this region of the recombinant fusion protein to be affinity chromatography separated. So you can use a nickel column or nickel matrix to purify the recombinant protein, because histidine can be purified by using divalent ion. So a stretch of 6 histidine can be used for affinity purification of the fusion protein. It contains this epitope here, express epitope, meaning that you can use an antibody for the detection and purification as well of the recombinant protein. But in our lab, we will not use that. We will only use the 6 hits for affinity purification. Afterwards is EK, which is called enterokinase site, meaning that you can have a protease that's specifically cleaved at the end of the EK. So you can get rid of this whole N-terminal fusion partner region. This whole region you can get rid of it by using an enzyme, enterokinase. But we won't do it in our lab. But in this vector, you have that already in place, just in case after you produce recombinant protein, get rid of the fusion partner. You can do that. After the EK, you have the multiple cloning site, MCS. Multiple cloning site is a stretch of DNA that contains unique restriction enzyme cut sites. It has to be unique. Otherwise, if you cut with BAMH1, if you cut the vector, then you destroy the vector. Now you only open up the vector at this particular region. If you cut with BAMH1, it will only open up here. But for us, we want to force clone our cDNA into BAMH1 and EcoR1 highlighted in this. So we will have a stretch of DNA that correspond to the 5' extracellular domain of the human secretin receptor. So it's a small part of the receptor. it's about 120 some amino acid long. So about 300 to 400 base pair of DNA. At the 5' end of this DNA, it is cut with BMH1. At the 3' end of this DNA is cut with equal R1. And the same is also happening in the vector. So you force clone the DNA that encodes the 5' extracellular domain of the human secretin receptor into the BMH1 and equal R1 site of this vector. So this double-strand DNA now is inserted into this BMH1 equal R1. And after the MCS, there's a stop codon. So we stop the translation process. Okay, so I repeat what I have said and summarize that. So this vector now is capable of producing a recombinant protein for you that contain an N-terminal fusion partner with the 6 times his tag. And then it contains also in the C-terminal region, the human secretin receptor 5' extracellular domain. So this is the result of what you have done in this genetic engineering project. Is that, you raised your hand. You have a question? Did you raise your hand? Sorry? No? Oh, you're just scratching your head. Any questions at this point? Because I have to explain more of the system. It's more complicated than that. Life is like this. Any question? You can raise your hand. Is that clear? Is that clear? You have any question? Okay, good. Okay, so this is the bacteria. This is the E. coli, the host. This is the PR set A with the DNA cloned into the BMH1 equal 1 site as shown here. And this is driven by the T7 RNA polymerase, right? RNA polymerase is an enzyme that drives transcription. But it will only recognize certain DNA sequence in front of a gene. It's called a promoter. It drives expression, meaning that it will transcribe that DNA, right? So this is the tRNA, T7 promoter that is recognized by T7 RNA polymerase. And the T7 RNA polymerase bind to this promoter and drive the expression of this fusion protein, drive the expression of this transcript RNA, messenger RNA, that encodes the fusion protein, right? So that part is, now you understand already. But we want to introduce an inducible system. So we don't want the protein to be expressed all the time. We want the protein to be expressed only if we want the protein to be expressed. Only when. Okay, so when the bacteria is growing in the culture, in the beginning, we do not want the protein to be expressed. These proteins being expressed early, they'll be degraded. They will be destroyed. And the bacteria is using all the energy producing these proteins. You don't want it. You want the bacteria at the beginning to use all the energy to replicate, to produce more plasmid DNA, so that it will grow in terms of size, in terms of size and number. Okay, so now you inoculate this bacteria in a solution, in a culture solution, put it into an incubator shaker overnight, and then this bacteria will divide, and divide. Now you have billions of this small little factory producing the protein for you, but you don't want it to do that. Until in the morning you come in, you start a fresh new buffer, you inoculate this buffer with this overnight culture, and then it will start to grow exponentially. Very nice new home for the bacteria, right? Fresh nutrients and fresh solution. So it will start to grow exponentially, and then we will stop the growth at the mid-log phase, when the bacteria is the most healthy, mid-log phase. Okay, so when the bacteria is healthy, we inoculate, we put some chemical into the solution, into the culture, to induce expression of this recombinant protein. And then we grow it for a couple of hours, 1 hour, 2 hours, and then we will harvest the protein. So we therefore introduce an inducible system. So this is the inducible system here. Okay, so the idea is to induce expression of T7 RNA polymerase only when we want the protein to be expressed. And how to do that is by another inducible system. And I'm sure you have heard of lag promoter and using lactose to induce the lag promoter. Have anyone learned that before? Lag promoter, lag operon. Have anyone? Can you raise your hand? None? Have you not heard of that? Can you raise your hand? Okay, so it doesn't matter. It's a very simple system. Anyway, so T7 RNA polymerase, this gene is here, and behind that, in front of that, it is driven by the lag promoter. The lag promoter is driven by the E. coli RNA polymerase. So E. coli RNA polymerase binds to the lag promoter to drive the expression of T7 RNA polymerase. But the lag promoter is controlled by the lag repressor, and the gene is here in the bacterial genome. The lag repressor normally will interact with the lag promoter to prevent the binding of the RNA polymerase onto the lag promoter, therefore stopping the expression of T7 RNA polymerase. Okay, so we put in a inducer, it is called IPTG. IPTG has similar structure as the lactose. So it can interact with the lag repressor, and then it will de-repress the lag repressor. It will change its conformations. And then now the lag repressor with IPTG is not capable of binding onto the lag promoter, is not capable of stopping RNA polymerase to interact with the promoter to produce T7 RNA polymerase. So by putting in IPTG into the culture during the mid-log phase, therefore you induce expression of T7 RNA polymerase, which will interact with the promoter to drive the expression of the target gene. So this is an inducible system that is normally used in all bacterial system to produce recombinant proteins. Is that clear? Good. So in your lab, what you will do is you will run, each one of you will run a gel like this. So you will collect, each one of you will, in the beginning, will be given a culture. And then after you spin down the culture, you homogenize. And then you open up the bacteria to release its content. And then what you will do is you will use a nickel matrix to separate the recombinant protein from all the other proteins. because it's now affinity chromatography. So nickel is able to, the matrix is able to interact specifically with the fusion protein because it contains a his tag. Okay, so you will collect the sample along your purification process. So supernatant 1, 2 flows through. You wash the matrix a couple of times, wash 1, wash 2, and wash 3. And this is the DNA size marker that you load onto the gel. So you know the molecular weight of each one of these proteins, as shown here in this diagram. And then you elute your protein. And this is the first one, first batch of eluent. And then this is the second elution of your fusion protein. You will see that you can purify your fusion protein almost to homogeneity, so up to 80, 90%, using only one simple nickel matrix. Okay, so each one of you should be able to produce a gel like that and write a full report based on that, on how you purify your protein. Okay. Okay, so chart to show you some of the, in the fusion protein, you can actually use anything, any fusion partner. So we talk about the his tag, hexahistidine tag, 6 times his, 6 amino acid long fusion partner. Many other fusion partners are being used in the biochemistry laboratories. You can use a MIG tag or HA tag flag. These are 8 to 10 amino acid long small peptide regions. So very simple to engineer into your fusion protein. So you can use this for detection, basically, for antibody detection. One I have used many times before is glutathione S transferase, GST. GST dramatically increases the solubility of the fusion protein. Sometimes when you produce too much protein, they will form what we call inclusion bodies. They are insoluble, difficult to purify them. So I use a GST partner before. Green fluorescent protein is one of the most popular protein tag nowadays because this protein is able to fluorescent. So it's a naturally fluorescent protein from jellyfish, and you can actually see the fusion protein because it is green color. You can use laser to activate and provide data. So you don't even need a laser to. Sorry, you don't even need a laser. So it will naturally fluoresce. And then, as you can see here in this, there are other tags that one can use as well. Okay, so now after your SDS page, this is a setup for Western blot. In fact, Western blot is a combined SDS page with antibody detection. So, so this is your gel here by electrophoresis. You separate your protein with the protein marker. And the gel you can take out. And then all you need to do is to put the gel on top of a nitrocellulose paper or nylon paper that you can purchase from the company. And then on top and below, you put some paper that is soaked with solution. And then you put this is the gel, this is nitrocellulose paper underneath. On top and below, you put in some filter paper soaked with solution. And then you put it into, like a sandwich, into this system here. When you apply a voltage from top to below, asking the protein to migrate from the gel onto the nitrocellulose paper. And then after the transfer, you take the paper out and you can bake it in a oven so that you cross-link the protein directly onto the nitrocellulose paper or nylon paper. And then afterwards, it's the same business like ELISA. You block with high protein concentration solutions. You detect with the primary antibody. It can be a direct detection if the primary antibody is conjugated with an enzyme. It can be indirect detection where the primary antibody is recognized by secondary antibody conjugate with an enzyme. And then you use a chromogenic substrate, a substrate that can generate color, so that you can generate a band. So now you get information on whether you have the protein in your sample. So if you see a band, that means you have the protein. And you can also measure the molecular weight of the protein by comparing to standards. And then you can tell people that this is what I want to identify because it has the same molecular weight as the one that I am trying to identify. Or you can tell people this protein has a molecular weight of like 60 kilodalton whatsoever. Okay, so the molecular weight is an information. Is some information they can get from Western blot. You can also compare the intensity of this band. This is what we usually do in our laboratory. Okay, so you can, for example, stimulate some cells, like liver cells with a hormone, and in the control without hormone. You run the gel alongside, you detect the protein. You can say that after protein stimulation, after hormone stimulation, then you can detect a higher concentration of a certain protein, meaning that the hormone is able to stimulate the expression of that particular protein. Okay, so things like that you can do. So it's a powerful technique. And if you want to publish your data in journals, this is like what you have to do nowadays. So as a Western blot. What time is it now? I take a short break here, and then we continue. Hopefully we can finish all the lecture material today.